The paper we are presenting on is called Molecular Mechanism of Bacterial HSP90 pH-Dependent ATPase Activity. It was written and performed by Yi Jin, Rael S. Hoxie, and Timothy O. Street. So there were a few things that the researchers already knew before they even performed the experiment, such as the nature of HSP90 to begin with, the general mechanism of HSP90, as well as the importance of studying HSP90. So they already knew that HSP90 is a chaperone protein that is a dimeric molecule, and it functions as a heat shock protein, aiding in the folding of proteins. So HSP90 stands for heat shock protein 90, with the 90 representing the atomic mass of the molecule in Dalton. So they also knew the general mechanism of HSP90, which is that it has a highly regulated open to close to open conformational cycle that is initiated by the binding of ATP and the hydrolyzing of ATP. So finally, the researchers also know that HSP90 is important to be studied because it is involved in various cellular processes, such as cellular regulation and cell proliferation. So this is crucially important because we know that those things are linked to the development of cancer. So the main goal of this experiment for the researchers was to identify the specific mechanism of bacterial HSP90, which is called HTPG. In order to further understand that mechanism, they were hoping to gain insights into human HSP90 and therefore hopefully make some breakthroughs in the research in that sense. So there were three critical parts to this experiment. So number one, knowing that HSP90 was a dimer, they chose to mutate the dimer in order to determine whether a salt bridge forming on one half of the dimer or both halves of the dimer is important in ATPase activation. So in mutating the dimers, they figured out that if you had two salt bridges and if you had one salt bridge, it was exactly the same result in either case as to how ATPase mechanism was activated. And they also found that if you had zero salt bridges, that ATPase was not activated. So the second thing that they determined was that ATP binds to a specific histidine at the position of 255 on the HSP90 dimer. So this is important because the binding of ATP to the 255 histidine results in a salt bridge forming, and that's what causes the ATPase activation. The third thing that they discovered was that by altering the pH, they could alter the pKa's of both the ATP and the histidine molecule, and that resulted in changes in the affinity of how the ATP bound to the H255, which therefore resulted in a difference in how ATPase activation occurred. So they discovered that ATPase activation is directly correlated to catalyzing activity of the HSP90 protein. And they discovered that if the affinity of ATP to the histidine molecule was lowered by changing the pH, then the ATPase activity and therefore the catalyzing activity was actually increased for the molecule.
Alright, so results for figure 3 show that there is an inverse correlation between nucleotide binding affinity and nucleotide or the HSP90 ATPase activity. So this particular experiment shows that there is a optimum pH range where the pK values are shifted. It's a broader range, and at around pH of 7, a little bit before 7, probably 6.6, there is a strong salt bridge between a histidine at 0.255 and on the amino acid chain that binds with ATP. This strong salt bridge stabilizes the open state, which is catalytically inactive, so there's no ATPase activity. The protein really doesn't fold the other substrate proteins that it's supposed to fold. However, though, this pH dependency shows as it moves away from that optimum pH. This means that there's a weaker salt bridge, which creates a higher activity that this protein kind of executes. So the protein does its job better whenever it is a lower salt bridge because this ATP will keep it kind of stabilized. And the way that this protein, the way that it kind of folds other proteins, it's a chaperone. So it assists other proteins in their late folding stage by going through its conformational cycle. And this is done through ATP binding and hydrolysis. So if there's a weak salt bridge between ATP and the histidine, it's able to go through its cycle more easily and do its job. So this one is just showing that there's an optimum pH where the ATP is stabilized. This graph, however, is showing an anti-correlation between catalysis and a binding affinity of histidine. So the higher the catalysis, the lower the affinity there is. So the lower the affinity for histidine, the higher the catalysis, which makes sense because if it has a high affinity to bind to histidine, then there's not going to be much catalysis of assisted protein folding.
So one of the results from the experiment can be seen in this figure here, where you have a visual demonstration of the relative positions of ATP and the histidine molecule relative to each other in the open and closed states of the dimeric molecule. So you can see that in the open position, the ATP is really close to the histidine molecule, and then as the dimer closes and ATPase occurs, the ATP is separated from the H255 molecule. You can also see in this diagram, well, there are three separate parts of the diagram. So in the first diagram, it's showing that relative to three other forms of HSP90, such as the human form and the yeast form, bacterial HSP90, also called HTPG, has a pH-dependent ATPase activity, which is shown here. After you reach a neutral pH, ATPase activity increases, whereas in other forms of HSP90, ATPase activity remains constant regardless of the pH change. So over here in this diagram, you can see that relative to ATP concentration, ATPase activity also changes, and then you have different pHs here as well. So the bottom here represents the lowest pH, and then as you increase pH, which is the color increases and changes, you would also increase ATPase activity. Finally, in this diagram, you can see that the catalysis rate of HSP90, that is bacterial, also increases after a pH of 7, and this shows the relationship between ATPase activity and catalysis activity, which is the same. As ATPase activity increases, so does catalytic activity, and this just represents the pH dependency of it.
Okay, so finally, let's conclude and figure out what we learned here today. So what we studied was the HSP90. It's a dimeric molecular chaperone. It assists proteins that are folding in their late stages that are involved in. The reason that it's important is because those proteins are involved in cell signaling and cell proliferation. So we studied this HSP90 family. Particularly, we studied the HTPG, bacterial HSP90, because we saw a pH dependency. This pH dependency is caused by an electrostatic interaction between a gamma phosphate and a histidine at 255 on the amino acid chain. The histidine is what is stabilizing the ATP and will kind of keep it in an area where it won't do its activity. And the transition between this salt bridge, between one salt bridge and no salt bridge, gives the energy that holds this, or gives the energy that it will need to overcome for the molecule to close. So it has a slow rate of closure compared to other rates of subsequent ATP binding and hydrolysis. The reason this is all important is because of the idea that we can re-engineer this bacterial HSP90 and involve it into cytosolic HSP90. So if we can re-engineer this and potentially put it into cytosolic HSP90, which is in humans, then there's the potential that we can inhibit certain cell proliferation and cell signaling that is involved in cancers. So the proteins that would get mutated and kind of lose control of their signaling and their proliferation, inhibitors of the bacterial HSP90 and what we could re-engineer would be very advantageous for us. So if we can inhibit these cell signaling processes and cell proliferation through this HSP90, then we're inhibiting those proteins that are involved in those processes. So all in all, the reason this is important is because it gives insight. We can learn into electrostatic mechanisms in certain chaperone proteins and move forward in the medical field in inhibiting and preventing cancers.